Thanks a lot, Bill. It's my privilege to finally bring some of the forbidden fruit of health effects into the discussion, and we'll see whether it's ripe or not quite so ripe. The first speaker is Ryan Allen. I don't think I've ever met Ryan, but I know his mentor very well, Joel Kaufman from the University of Washington. Ryan has trained as an exposure scientist, an epidemiologist, and as a health effects person. And was first author on what I think of as one of the seminal papers looking at health effects as a function of an air cleaner intervention. So anyways, without further, he's now at Simon Fraser University, and hopefully he's hearing me on the ether, and he will pick up from where I leave off. Ryan?
Okay, good morning, everyone. I'm assuming everyone can hear me, so I'll just jump right in unless I have some indication otherwise. So it's a pleasure to participate in this meeting. I'm sorry I couldn't be there in person today. I had another event yesterday that prevented me from traveling. But as was just said, I've been asked to speak about indoor particulate matter and health, specifically cardiovascular health. And so here's what I want to try to cover in the next 20 minutes or so. I want to say a little bit about our current understanding of PM and cardiovascular health. So before I jump in and think about indoor PM, I want to say a few words more generally about what we know about PM and cardiovascular health, because as you all discussed yesterday, there are obviously important differences between indoor and outdoor particles, but what we know from studies using outdoor measurements and models still has some relevance to our discussion of indoor PM. Then I'll shift into the indoor air studies, and I've organized this sort of as the study designs that are used to evaluate indoor air and cardiovascular health, including in vitro studies, so-called panel studies, including a subgroup of those panel studies, which are the intervention studies that Howard just mentioned, and then cohort studies, or given the relatively limited state of this literature, instead of cohort studies, it's actually cohort study, singular. I'll finish with a few what I see as kind of the knowledge gaps in this area, and I think there are several important knowledge gaps. I'm not going to cover household air pollution from solid fuel combustion, which, as most of you probably know, is arguably the most important environmental risk factor globally, but isn't really relevant in high-income countries like the US or Canada. I'm also not going to talk about environmental tobacco smoke. I sort of debated with myself about whether to include some of that literature, and I've decided not to, primarily in the interest of time, but we could certainly discuss it if there's interest. So I want to start actually by reiterating something that I'm sure you talked about yesterday. I wasn't able to participate, but I'm sure you talked about the contribution of outdoor particles to indoor environments. And the categorization of particles into indoor and outdoor is useful in many ways. Certainly the policy and the regulatory implications are very different. But I've always found that categorization slightly inconvenient and slightly almost misleading, because, of course, much of the quote-unquote indoor PM that we breathe is actually outdoor PM. So just to illustrate that point, these are results from the Mesa Air study that I participated in as a postdoc. And in that study, as part of that study, we have indoor and outdoor measurements in over 500 homes, 500 non-smoking homes in 7 U.S. cities. And with those indoor and outdoor measurements, we're able to estimate infiltration and estimate the contribution of outdoor generated PM 2.5 to the indoor concentration. And in these homes, we estimated that about 80% of the indoor PM 2.5 on average was actually of outdoor origin. And certainly if you looked in smoking homes, or if you looked at ultrafine particles or other components of the PM mixture, you might get a different picture. But the point is that when we think about indoor particles, we shouldn't ignore outdoor particles, and that the line between indoor and outdoor PM might not be quite as distinct as we may think. So as you probably know, a lot of the early research on air pollution and health, on PM and health, focused on respiratory health. Of course, the respiratory system is kind of the portal of entry for particles, and it's the way that our bodies encounter these particles. But since I got involved in air pollution research about 15 years ago, it's safe to say that, at least in terms of the epidemiology, the kind of hottest and most active area of research has actually been on the cardiovascular effects of PM and other air pollutants. This slide just shows screenshots of a number of review papers and editorials that have been published on this topic, and of course there are many more individual studies. But this is a very active area of research, and as you know now, people have moved into other areas as well, including some of the health effects that we'll hear about in the next presentations in this session. So we've learned quite a lot from that research, and one of the important things and one of the important conclusions that we've drawn is that these are not just statistical associations. These are actually causal relationships. This is a quote from an American Heart Association scientific statement that was published in 2010, 6 years ago now. But even at that time, the writing group was comfortable calling the relationship between PM 2.5 exposure and cardiovascular morbidity and mortality a causal one. Now, of course, this evidence base was almost entirely based on studies using outdoor measurements and models, but it is worth noting that 6 years ago we were already calling this a causal relationship, and of course more evidence has developed in the 6 years since. Another important kind of finding and conclusion may be too strong at this point, but certainly evidence is suggesting some interesting results in terms of the effects at low levels and the shape of the dose response or the concentration response relationship, where we're seeing effects down really to the lowest levels that people encounter, down to just 5 micrograms per cubic meter of PM 2.5 or even lower. And that suggests, and the evidence seems to suggest, the absence of a threshold or the absence of a safe exposure level. Related to that is the idea that the relationships between PM and cardiovascular health outcomes seems to be linear or even supralinear, where we get the steepest the curve is steepest at the low end of the exposure distribution, and that has important implications, suggesting that even if levels are already low, you potentially get some health benefit from even further reductions in PM exposure and PM concentrations. So when you put all of that information together, you get what is actually a very important public health risk factor, and this is a figure from the most recent iteration of the Global Burden of Disease assessment, where they look at various public health risk factors and try to sort of rank them to get a sense of their relative importance based on disability-adjusted life years, which takes into account both morbidity and mortality. And we see that ambient PM is an important global health risk factor, as you already know. That's actually not why I'm showing you this figure. The reason I'm showing you this figure is that it turns out that the public health impacts from PM are actually driven largely by PM's effects on cardiovascular and circulatory diseases. So and this is a result from a global assessment, but we would get exactly the same picture if we look just at the U.S. or just at Canada or just at other high-income countries. So we tend to think of in many cases, we still tend to think of PM as a respiratory pollutant or a respiratory toxicant, and it is, but it actually turns out in terms of its public health impacts, it's the cardiovascular and circulatory effects of PM that are really driving the public health burden from PM, and that's important to keep in mind. So this is a very busy figure from that American Heart Association statement that I mentioned a few minutes ago, and it summarizes the hypothesized mechanisms linking PM with effects beyond the lung, cardiovascular effects, as well as some of the other systemic effects that we'll hear more about in a few minutes. And I'm showing you this for two reasons. One, I want to convince you that it is plausible that particles can have these effects beyond the lung. But I also want to emphasize the importance or the way that some of these intermediate health indicators fit into this bigger picture. As I'll mention in a few minutes, most of the indoor PM studies can't look at clinical outcomes like heart attacks and strokes or the development of cardiovascular disease. So we're forced to look at more subtle kind of subclinical indicators of health, and so I want you to have some sense of how those fit into this bigger framework. So just very quickly to sort of summarize the key points in this very busy figure, there are essentially three interrelated mechanisms that we think link PM exposure with cardiovascular health. Number one here has to do with an inflammatory response. So we breathe particles into our lung. We get an inflammatory response in the lung. Some of that inflammatory response spills over, so to speak, into the systemic circulation, and that can have effects on blood vessels and on the blood itself. Number two here has to do with the autonomic nervous system and the fact that when we are exposed to particles, it may actually affect the autonomic nervous system's balance and affect the blood vessels as well as heart rhythm, heart rate variability, and those types of things. And then number 3 here has to do with the smallest particles actually making their way into the bloodstream directly and sort of directly interacting with our blood vessels and our blood. But I want to emphasize a few kind of pieces of this very busy figure. One is that we can look at various cytokines or various proteins as indicators of this inflammatory response, and a number of the indoor PM studies have done that. We can look at blood vessel function, basically how well blood vessels expand or contract as a result of changes in blood flow. We can look at changes in blood pressure, and we can look at changes in indicators related to heart rate or heart rate variability. And all of those outcomes are relevant given what we think are the biological mechanisms at play here. So the methods that have been used then to investigate indoor PM in health include in vitro studies as well as epidemiologic studies, as I said. The epidemiologic studies in the context of indoor PM have some really important limitations. And really the key idea here is that if we're going to understand indoor PM, we've got to measure indoor PM. We're still, I would argue, not all that good at modeling it. There have been some good attempts, but we're still not developing really great models for predicting indoor PM. So in most cases, we have to measure it. And what that means is because these measurements are time-consuming and take effort and take money, we end up studying relatively small study populations that may actually be non-representative. So we may have issues in terms of extrapolating our results to the broader general population. We're typically limited to studying short-term effects that occur on the order of days or weeks, which is a limitation because we now know that many of the effects of outdoor PM, ambient PM, are chronic and take place over many years. And related to that, we're only looking at relatively subtle subclinical effects, the types of intermediate markers that I showed you a few minutes ago, and we're really not able to look at kind of clinically relevant outcomes, heart attacks, strokes, and those kinds of things. So there are some limitations in terms of what we're able to do with the epidemiologic research. Just to kind of illustrate this idea of these in vitro studies, I pulled up a study that's now 15 years old. It was one of the kind of early looks at this, and it was kind of a clever idea by the Harvard group. They looked at PM samples that had been collected indoors and outdoors at Boston area homes, and they exposed cells to these particles, in particular rat alveolar macrophages. So macrophages are sort of the police that kind of patrol our lungs and look for invaders. And so they exposed these cells to these different particles collected indoors and outdoors, and looked at the inflammatory response from these cells using a particular cytokine related to inflammation. And they found in this small kind of pilot scale study that they actually got a larger inflammatory response from the indoor PM than from the outdoor PM. That was largely due to differences in endotoxin, but even when they normalized for endotoxin content, they still saw a greater inflammatory response from the indoor particles from some other component of the PM. Now, I don't want to kind of overemphasize this or overinterpret this. This is one study that I'm showing you really just as kind of an example, but it's one tool that we have available to us to potentially try to understand indoor PM and kind of the differences between indoor and outdoor PM in terms of inflammation or other relevant health indicators. But what I really want to focus on for most of the time that I have left is on these intervention studies, these air filter studies that Howard mentioned at the outset. These studies use an air filter to reduce PM concentrations indoors, and one of the advantages of that approach is that you reduce the particles much more than you reduce the gases or the other co-pollutants. So it's kind of a way to really sort of isolate the effects of PM from all the other stuff that may be correlated with the PM. Most of these studies have used a randomized crossover design. So this is a figure from a study that we conducted in British Columbia in a wood smoke impacted community in the central part of the province, where each participant got 2 weeks of monitoring. At the end of the first week, we did some health measurements, and then we came back at the end of the second week and repeated those health measurements. One of the weeks, the filter was in operation. The other week, the filter was not in operation. It was actually a filtration unit without the filter inside of it, so we call it kind of placebo filtration. And participants were randomly selected to either get the HEPA filter first or the placebo filtration first, and that's the randomized crossover piece of this. And this is a useful design because you end up comparing people to themselves. You end up comparing an individual to him or herself, and that has benefits in terms of reducing the potential for bias from other things that might be related to these outcomes. And these studies, another advantage of these studies is they sort of allow you to ask the question, what would happen if we reduced PM concentrations? So this is the first study that we did. This is the one that Howard referenced in when he was introducing me. So the take-home message here is that we found that the use of these portable air purifiers for 7 days in these homes was associated with improved blood vessel function and decreased systemic inflammation. So RHI is reactive hyperemia index. It's a measure of how well blood vessels respond to a change in blood flow, and then we saw decreases in C-reactive protein, which is a protein related to inflammation. So we know that the air filters affect exposure. In this case, the PM 2.5 concentration was reduced from about 11 micrograms per cubic meter to about 5 micrograms per cubic meter. We know that these filters work in terms of reducing PM. The question was, do they have any benefits in terms of health? And based on this study, it seemed to suggest there were some benefits in terms of some of these intermediate cardiovascular health markers. We followed that up with a study that was essentially the same design but with one wrinkle. The question we were interested in here was, do the effects of indoor PM on cardiovascular health indicators vary depending on the kind of main source of PM at that location? And the two main combustion sources of PM that we worry about in Greater Vancouver are traffic emissions and residential wood combustion. And so using some previously developed spatial models, we were able to identify parts of the city that were impacted by traffic-related air pollution but not wood smoke. And not surprisingly, those were areas near major roads that are shown in red. And then areas that were impacted by wood smoke but not by traffic, and those were shown in green. And so the idea here was to use the air filter to introduce an exposure contrast or an exposure gradient that was comparable between these two groups. So one of the challenges to trying to understand different sources, as you know, is that the exposure variability and therefore the ability to see associations with health can be different for PM from different sources. So in this case, the air filter allowed us to introduce a gradient that was similar in magnitude for both groups. And what we found here was that there was an association between indoor PM 2.5 and C-reactive protein, but we only found it among the group that was impacted by traffic and not among the group that was impacted by wood smoke. So, you know, the take-home message here is really something that you probably all already know kind of intuitively, and that is that the indoor PM and its relations with health outcomes depend on where the PM 2.5 is coming from. In this case, you know, this one small study suggesting that maybe traffic particles may have some greater impacts on inflammation than wood smoke particles. There have been a number of these air filter intervention studies published over the last 6 or 7 years, and the results are more or less consistent, so I'll just try to very quickly summarize them. The first study that was actually published a little bit before ours, so it's probably the seminal study in this area, was by Browner and colleagues, where they looked at people in Copenhagen who were exposed primarily to traffic emissions, and they found that use of indoor filters increased blood vessel function, and interestingly, actually found that the PM 2.5 was a better predictor of the health response than the particle number count or the ultrafine particles. So that's kind of interesting. This group followed up that study in Copenhagen 5 years later, when levels in Copenhagen had come down quite a bit. They didn't see an effect with the filter itself, but they found that the PM 2.5 concentration as a continuous variable was associated with reduced reduced blood vessel function using that same indicator of blood vessel function. There was a study in Taiwan where levels are obviously a bit higher, and they found that there was an association between PM2.5 and both blood pressure and heart rate, but only during the period when the filter was not operating. So when the filter was turned on, they didn't see these relationships quite as clearly, but with the filter off, they saw these links. Scott Weichenthal published a study based on Canadian data among a native Canadian population in Manitoba, and they found that filtration was associated with a reduction in blood pressure. In this case, there was a lot of environmental tobacco smoke in these homes, so that study is really effectively an environmental tobacco smoke study, but nevertheless, they did see some links with blood pressure. This study in Massachusetts is probably the study where the results fit in not quite as well as some of the others. The results are a little surprising and inconsistent. They found that filtration actually increased IL-6, which is a cytokine related to inflammation, and they found that particle number count increased IL-6, but was also associated with a decrease in blood pressure. So the direction of some of the effects were not in the direction that we might expect. And then most recently, there was a study in Shanghai where, again, the levels would be considerably higher, but they found that filtration was associated with reduced inflammatory and thrombogenic biomarkers, as well as reductions in blood pressure. So generally speaking, the results are consistent. We see that the filters or the PM2.5 exposure reductions associated with the filters may lead to changes in blood vessel function, changes in inflammation, and potentially even changes in blood pressure. So the last study I want to tell you about, I'll try to finish up here quickly so I don't go over my 20 minutes, is really the only cohort study that I'm aware of. As I mentioned, we have some challenges when we're limited by indoor measurements in terms of what we can study and how long we can study. This study is different in the sense that the exposure assessment was a questionnaire. So this is incense use and cardiovascular mortality among Chinese in Singapore. And long story short, they found that incense use daily for over 20 years compared with no incense use was associated with about 10% increased risk of cardiovascular disease mortality and coronary heart disease mortality, and about a 20% increase in the risk of stroke. So although it's probably not an exposure incense use that is particularly relevant in North America, it does provide some evidence that particles generated indoors and if exposure is chronic, can have some important implications for actual clinical outcomes like mortality. So then just to summarize here, we know PM is an established cardiovascular risk factor, but most of that evidence comes from studies of outdoor measurements or models. And the public health impacts of PM are actually driven by its cardiovascular effects. So from a public health point of view, these cardiovascular effects are very, very important. This study of indoor PM presents some important epidemiologic research challenges. As I talked about, when we need to measure indoor PM, things become more difficult. And although not entirely consistent, human studies have reported links between indoor PM and increased systemic inflammation, impairments in blood vessel function, and to a lesser extent or less consistently increases in blood pressure, which all fit in kind of mechanistically with what we think is happening between particles and cardiovascular health. And finally, just to conclude, what I see as some important knowledge gaps, I think we still don't have a great understanding of the relative toxicity of indoor generated versus outdoor generated PM that's found indoors. We still don't have a great handle on the relative toxicity of PM from different sources and the relative toxicity of PM 2.5 mass concentration versus ultrafine particle counts. We don't have a great handle, I would say, on the role of bioaerosols on the cardiovascular effects of indoor PM. And because of some of the research limitations that I mentioned, we don't have a great handle on the relationship between long-term exposure and the more clinical outcomes like mortality, heart attack, and stroke. So with that, I'll conclude, and I thank you for your attention, and I look forward to the discussion period. Thank you very much.